The deadliest poison known to human science isn't cooked up in some high-tech clandestine military bunker.
Right, not at all.
No, it's actually sleeping in the dirt of your backyard right now. So, welcome to this deep dive, custom tailored just for you. Today, we are immersing ourselves in a microscopic world defined by, well, staggering paradoxes. We're focusing on a duo we're calling the Toxin Twins.
I love that name for them.
Yeah, it fits perfectly. We're looking at two virtually indestructible organisms that are responsible for some of the most agonizing diseases in human history.
Yeah.
But at the same time, they basically laid the foundation for modern immunology.
Yep.
They spawned a multi-billion-dollar cosmetic industry, and they're classified as top-tier bioterrorism threats. Like, millions of people willingly pay to have one of these lethal toxins injected directly into their faces every single year.
The contrast is just absolutely phenomenal. I mean, we are dealing with ancient soil dwellers that are entirely indifferent to human existence.
They don't care about us at all.
They don't need us to survive. Yet they possess this biological toolkit that is so remarkably precise and, so devastating to the human nervous system that we have spent the entire last century locked in this strange dance with them.
A very deadly dance.
Exactly. Alternately trying to eradicate them from our food and our wounds, and then on the flip side, trying to harness their power for medicine.
Okay, let's unpack this. Before we get into the cosmetic empires and the biological weapons, we really need to understand the biological foundation of these microbes.
The basic building blocks.
Right, like what they actually are and the terrifyingly efficient logic that they share. We're talking about Clostridium botulinum and Clostridium tetani.
So, from a biological standpoint, these two belong to the phylum Firmicutes. They are anaerobic, spore-forming, gram-positive bacilli that...
Wait, I have to pause you right there.
Yeah, sure.
I am a deeply curious person, but you just threw a whole microbiology textbook at me in like one sentence. Let's break that down for you listening, especially if you haven't been in a lab recently. Gram-positive bacilli. What does that actually mean?
Fair enough, yeah. So bacilli just refers to their shape. They look like microscopic rods.
Like tiny hot dogs.
Yeah, exactly like tiny hot dogs. And gram-positive refers to the structure of their cell walls. They have this very thick outer layer that holds onto a specific purple stain used in labs.
Oh.
It just helps scientists quickly categorize them when they're looking under a microscope.
Got it. And what about the anaerobic spore-forming part? Because our sources highlight that as their, ultimate survival strategy.
That is the key to their entire existence.
Hmm.
Anaerobic means they cannot tolerate oxygen.
At all.
No, normal atmospheric oxygen is actually toxic to them. So to survive in our oxygen-rich world, they spend most of their time not as active growing bacteria, but as dormant spores.
Oh.
Think of a spore as a nearly indestructible armored seed. They sit in the soil, in household dust, or even in animal guts, just in this state of suspended animation.
Wow.
They can survive boiling, freezing, radiation. They just wait.
They just wait. And they will only wake up or germinate, right, when two very specific conditions are met.
Right.
No oxygen and lots of nutrients.
That combination is their absolute trigger. So a deep sealed puncture wound, or maybe a poorly processed can of vegetables, or the undeveloped colon of a newborn baby.
Which is terrifying.
It is. These are perfect oxygen-free havens. Once they find that environment, they shed their armor, they become active vegetative bacteria, and they start secreting what are known as AB exotoxins.
Okay.
Specifically, these toxins are zinc-dependent metalloproteases.
Okay, another big term. Metalloprotease. Does that mean they're, like, literally using metal to cut things apart inside us?
Pretty much, yeah. They use a zinc ion as the core component of a biological pair of scissors. It's an enzyme. Both of these twins produce a toxin that aggressively targets a very specific set of proteins in our neurons called snare proteins.
Now, this is where I want to test an analogy on you, because I was trying to visualize this shared molecular trick based on the research.
Let's hear it.
The best way I can picture it is by imagining a massive cargo ship trying to dock at a busy port. In this scenario, our neuron's synaptic vesicle, which is like a little bubble packed full of vital chemical neurotransmitters, that's the cargo ship.
Okay, tracking so far.
And the snare proteins are the heavy essential docking ropes needed to tie the ship to the pier so it can open its doors and unload its cargo to send a signal to the next nerve.
The mechanics of that analogy hold up perfectly, actually.
Okay, awesome. So the vesicle comes in to dock, but the zinc-dependent toxin acts like a pair of rogue shears. It just cuts the docking ropes, Exactly. The ship is right there at the pier. The cargo of neurotransmitters is ready to go. But because the snare proteins are severed, the vesicle cannot fuse with the membrane. It literally cannot unload.
Right.
The chemical signal is completely silenced. No matter how frantically the brain tells that nerve to fire, nothing happens.
When you look at the implications of that severed rope mechanism, you really start to understand their devastating potency. I mean, most bacterial infections need to overwhelm your immune system through sheer numbers.
Right, an army.
Yeah, an army. But here, a microscopic, localized population of bacteria, maybe just hanging out in a millimeter-wide wound or like a single spoonful of soup, can secrete enough of this enzyme to completely incapacitate a host that is millions of times its size.
Unbelievable.
The enzyme just keeps cutting rope after rope, disabling nerve after nerve.
But that raises a massive question, a massive why. If these bacteria are just soil dwellers that don't need humans to survive, why on earth did they evolve a neurotoxin perfectly calibrated to destroy human nervous systems?
It is a brilliant, albeit morbid, evolutionary strategy. The thing is, they aren't targeting humans specifically.
Oh, they aren't.
No, they're targeting animals and insects. Remember, these bacteria need an oxygen-free, nutrient-rich environment to grow.
Right, the trigger.
What better way to create that environment than to paralyze a passing animal? The animal dies, its body decomposes, the oxygen is depleted by other decay bacteria, and suddenly you have a massive, localized anaerobic feast for the Clostridium spores to wake up and multiply in.
Wow, so we are just like collateral damage in their quest to turn living things into rotting, oxygen-free buffets.
That is exactly what we are.
But this brings up a huge fork in the road regarding how they affect us if they use the exact same molecular scissors, right? If they're cutting the exact same docking ropes in our nervous system, why do the resulting diseases look so incredibly different? Botulism and tetanus are clinical opposites.
What's fascinating here is how one microscopic change in neurological targeting shifts the entire clinical presentation from total limpness to rigid, bone-breaking spasms.
It's wild.
It all comes down to their travel itinerary within the nervous system.
Okay, I know botulinum acts locally at the neuromuscular junction, right? It blocks the release of acetylcholine, the neurotransmitter that tells your muscles to contract.
Correct.
So the muscle gets absolutely no signal, so it goes entirely limp. You get flaccid paralysis.
The signal from the brain simply hits a brick wall. But tetanus has a much more insidious route. The tetanus toxin enters the motor neurons at the wound site, but it refuses to stop there.
Oh, it keeps going.
Yeah, it hijacks the neuron's internal transport system and travels backward up the axon of the nerve. It's a process called retrograde axonal transport. It literally climbs the nerve all the way into the spinal cord.
So it gets into the central nervous system. What does it do once it's inside the spinal cord?
It crosses over into adjacent inhibitory interneurons, specifically a type called Renshaw cells. These cells use neurotransmitters called glycine and GABA.
Wait, are those the body's natural brakes, like the signals that tell a muscle it's okay to relax?
Yes, they are the vital dampeners of our nervous system. So while botulinum cuts the accelerator cable, tetanus destroys the brakes.
Oh, wow.
Without glycine and GABA to send that relax signal, your motor neurons begin to fire uncontrollably at maximum capacity.
Which completely shatters the assumption that similar toxins cause similar symptoms. Tetanus gives you violent spastic paralysis. The muscles lock up entirely.
Yeah, it's horrific.
You get trismus, commonly known as lockjaw, because, well, the jaw muscles are among the most powerful in the body and they seize first. Then you get those severe arched back spasms called opisthotonus, and a rigid forced grimace called rhizus sardonicus. I read that the muscles contract so violently that patients can actually tear their own tendons or fracture their own bones just from the internal force.
That's entirely true. The clinical manifestations are polar opposites. And interestingly, that stark divergence in symptoms is mirrored by how radically different their historical paths to discovery were.
Right, because if these two microbes look identical under a microscope but cause completely opposite diseases, how long did it take humanity to realize they weren't the same thing?
Well, for tetanus, the mystery was unraveled through rapid, highly focused lab science in the late 19th century. In 1884, a German scientist named Arthur Nicolaier injected soil into animals, and proved this spastic paralysis was caused by a soil-borne microbe.
Okay.
But he couldn't isolate the bacteria itself because the oxygen in his lab kept killing his samples.
I can imagine how frustrating that must have been for early bacteriologists. They knew it was there, they could literally see the effects, but they couldn't grow it.
It was incredibly frustrating. It actually took a brilliant Japanese bacteriologist named Kitasato Shibasaburo to crack it in 1889.
Ah.
He was working in Robert Koch's famous lab in Berlin. Kitasato realized he had to eliminate the oxygen entirely, so he grew the cultures under a bell jar filled with hydrogen gas.
Oh,
Very. And then he applied severe heat. He knew that tetanus spores could survive boiling temperatures that would destroy all the other contaminating vegetative cells in the sample. Through that combination, he finally isolated a pure culture of C. tetani.
And the momentum didn't stop there. The very next year, 1890, Kitasato and his colleague Emil von Behring figured out passive antitoxin immunization.
Yes.
They discovered they could take blood serum from an animal that had survived the toxin and inject it into another animal to cure it. I mean, that single discovery essentially birthed modern immunology and won von Behring the very first Nobel Prize in Medicine in 1901. It was like a blitzkrieg of scientific progress.
It really was.
But the story behind botulism, that is entirely different.
Oh, completely. Botulism is a much older, much stranger narrative. It wasn't solved by a rapid sequence of laboratory breakthroughs. It was rooted in a very specific, terrifying form of food poisoning.
This part of the research completely blew my mind. We have to talk about Justinus Kerner in the early 1800s.
The sausage poet.
Yes. He was a German physician, but he was also a published poet. Over the course of 2 decades, he meticulously cataloged over 200 cases of what he called sausage poisoning Wurstvergiftung. He noticed people were eating poorly prepared blood sausages and then slowly going limp, losing their vision, and suffocating.
It was a very grim way to go.
And what's incredible is that decades before germ theory was even established, Kerner correctly deduced that this was a biological poison developing in the anaerobic center of the sausage, not some chemical contaminant like lead or arsenic.
His dedication was unparalleled. I mean, he even went so far as to extract the toxic juice from spoiled sausages and feed it to animals.
Which is wild enough.
But when that wasn't enough, he applied drops of the toxin to his own tongue to document the localized paralysis it caused.
Just casually putting the deadliest poison on his tongue.
Exactly. He laid the entire clinical groundwork for the disease. In fact, the word botulism is a direct homage to his work. Botulus is Latin for sausage.
But it took nearly a century after Kerner's self-experimentation to actually find the culprit, right?
Yeah, it wasn't until 1897 that the microscopic assassin was identified, following a tragic event in the Belgian village of Elazels. 34 local musicians performed at a funeral and shared a meal of raw, salted ham.
Oh, no.
Almost all of them fell ill with severe flaccid paralysis, and 3 died. A bacteriology professor named Emil van Ermingen was brought in to investigate.
And he managed to isolate our spore-forming twin from both the leftover ham and the victims.
He did. And crucially, he proved that it was a secreted toxin causing the illness, not the bacteria actively invading the victim's tissues.
Right, the bacteria just made the poison.
Exactly. The bacteria simply treated the salted ham as an oxygen-free incubator to produce the poison before it was ever consumed. His methodology is still taught today as a masterpiece of outbreak investigation.
So connecting those 19th century sausages and salted hams to the modern era, how do these organisms operate in the world today? Our sources outline 3 distinct modern scenarios that show how these twins still manage to terrorize us. The first is the 1971 Bon Vivant Vichyssoise outbreak.
That event permanently altered the American food industry. A single improperly processed can of Vichyssoise soup, which is a potato and leek soup, killed a man in New York and left his wife severely paralyzed.
It's just from 1 can of soup.
1 can. The subsequent FDA investigation triggered a massive recall that bankrupted the century-old Bon Vivant company in a matter of weeks.
That panic led directly to the incredibly strict FDA commercial canning standards we rely on today. They instituted what the industry calls the botulinum cook, which requires a 12-log reduction.
Right.
Which means, statistically, they have to heat the food to such an extreme temperature for such a specific duration that if there were 1 trillion botulinum spores in a can, only 1 single spore would survive. They made the math practically foolproof.
Yet, while we solved commercial canning, science recognized a completely new vector in 1976, infant botulism.
Ah, yeah.
This occurs when babies under 1 year of age ingest C. botulinum spores directly. The most widely known culprit for this is raw honey.
Now, if adults eat a few spores and honey, nothing happens, right? Because our mature gut microbiomes easily outcompete the dormant spores.
Yes, that adult microbial defense is incredibly robust.
Yeah.
But an infant's digestive tract is like a freshly tilled, unplanted garden. There's no competition.
Oh, that makes sense.
So the spores find a warm anaerobic environment. They wake up, colonize the colon, and begin producing the toxin right there inside the baby's body. It results in what's called floppy baby syndrome. They develop a weak cry, feed poorly, and lose head control.
Which is precisely why pediatricians are so adamant that infants should never consume honey before their first birthday.
Exactly.
Never. The third modern anchor point highlights a massive disparity in global health, specifically looking at wound infections. In wealthier nations, we often see wound botulism isolated among populations who inject black tar heroin.
Right.
The acidity of the drug and the tissue damage from the needle create these perfect oxygen-starved pockets under the skin for spores to germinate. But on the global stage, the tetanus side of wound infection is a heartbreaking story about neonatal tetanus.
It really is.
Yeah.
Historically, hundreds of thousands of newborns died every single year worldwide due to unhygienic umbilical cord practices. If a traditional birth attendant cuts the umbilical cord with an unsterile tool or packs the stump with soil or ash, which was actually a common traditional practice in some regions, they introduce tetanus spores directly into the wound.
Which is a perfect anaerobic environment.
Yes. The infant develops severe spasms and usually cannot survive.
But there is a massive silver lining here, thankfully. The World Health Organization launched an aggressive initiative to provide tetanus toxoid vaccinations to pregnant women in high-risk areas.
A brilliant strategy.
Yeah. The mother develops antibodies and passes them across the placenta to the baby, protecting the newborn during those vulnerable first few weeks of life while the umbilical stump heals. That single targeted intervention has driven neonatal tetanus deaths down by well over 90% since the 1980s.
It remains one of the most under-celebrated triumphs in the history of public health, honestly. But as we look at how well clinical medicine has learned to manage these diseases, we really have to acknowledge a massive identity crisis happening in the laboratories right now.
Oh, you mean the taxonomy?
Yes. The taxonomy, the way we classify and name these bacteria, is essentially broken. C. botulinum isn't a single species of bacteria at all.
Wait, what? It's a lie.
It's a taxonomic lie, yes. It is a functional category stitched together across at least 4 distinct genetic groups. If you analyze the DNA, Group I and Group Sorvi are more genetically distant from each other than many bacteria that exist in entirely separate genera.
Wow.
The only reason we group them under the name botulinum is that they all happen to produce this specific neurotoxin.
But the sources say the gene for this toxin doesn't even inherently belong to the bacteria's main chromosome. It's carried on a mobile bacteriophage.
Right, a bacteriophage is a virus that specifically infects bacteria.
Okay, so let me get this straight. The bacteria itself gets infected by a virus, and that viral infection gives the bacteria the genetic code to make the toxin that kills us.
That is horizontal gene transfer in action. It is a piece of row genetic code jumping between completely different microbes in the soil. You even have strains of completely different bacterial species, like Clostridium butyricum, picking up this toxin gene and causing botulism, even though they aren't botulinum at all.
That is insane. So if the taxonomy is completely phylogenetically wrong and the genetics are this chaotic, why doesn't the scientific community just rename it? I mean, they literally just renamed C. difficile to Clostridioids a few years ago to be more accurate. Why not fix this one?
If we connect this to the bigger picture, it comes down to a harsh clash between medical utility and phylogenetic reality. Put yourself in the shoes of an intensive care doctor staring at a patient whose lungs are failing due to flaccid paralysis.
Okay.
That physician only cares about one question. Does this patient have botulism? They need to know immediately so they can administer the life-saving antitoxin.
Right. Time is of the essence.
Exactly. They absolutely do not care which specific genetic genome or viral phage is producing it. If microbiologists fragmented the name into a dozen new, highly accurate species, it would offer zero diagnostic benefit to a dying patient. But it would create dangerous, potentially fatal confusion in hospital labs and public health reporting. Clinical practice has vetoed the microbiologists. The name stays, regardless of the genetics.
This tension between what a toxin is on a molecular level and how we interact with it in the real world leads us to the most famous and, frankly, most misunderstood aspects of the twins. We have to talk about cosmetics and biological warfare.
It is the ultimate duality of the natural world. In the 1970s, an ophthalmologist named Alan Scott began experimenting with highly purified botulinum toxin A. He used it to treat strabismus, which is crossed or misaligned eyes, by injecting microscopic amounts into overactive eye muscles to selectively weaken them.
And then, in the late 1980s, Canadian ophthalmologist Jean Carruthers and her dermatologist husband Alistair noticed a very interesting side effect. Patients receiving these eye treatments were losing their glabellar lines, the deep frown lines between their eyebrows.
And Botox empire was born.
Just like that.
Yep. Today, it's one of the most widely performed cosmetic procedures globally. But beyond aesthetics, it has profound therapeutic applications. It is used to manage chronic migraines, severe neck spasms known as cervical dystonia, excessive sweating, and overactive bladders.
Here's where it gets really interesting to me. The CDC classifies botulinum toxin as a Category A bioterrorism agent, putting it on the exact same threat tier as anthrax and smallpox.
It's no joke.
It is widely cited as the most acutely poisonous substance known to science. The lethal human dose is measured in nanograms per kilogram. The Aum Shinrikyo cult, the terrorist group responsible for the 1995 sarin gas attacks in Tokyo, they actively tried to weaponize botulinum toxin as an aerosol in the early 90s.
Which is a terrifying thought.
Very. We have the most lethal substance ever discovered by human science. And every single year, millions of people willingly pay hundreds of dollars to have it injected directly into their faces at strip mall clinics.
Their failure to cause mass casualties in the 90s wasn't because the toxin lacked potency, though. They failed due to complex engineering problems with mass dispersal.
Right.
But the cosmetic irony you mentioned perfectly illustrates the foundational maxim of toxicology. The dose makes the poison.
Ah, yes.
The exact same molecule is a weapon of mass destruction at a systemic dose and a safe, effective medical treatment at a localized microgram dose.
Speaking of things that are profoundly misunderstood, we need to clear up some major misconceptions before we finish. The biggest one has to be the rusty nail myth regarding tetanus. If you ask anyone on the street, they will tell you that rust causes tetanus.
It is an incredibly persistent myth. The iron oxide, the rust itself, is completely biologically irrelevant.
Wow.
The danger arises because a rusty nail often causes a deep, penetrating puncture wound, and is usually found outside, contaminated with dirt. It's the anaerobic, oxygen-free pocket created deep inside the tissue by the puncture, combined with the presence of soil spores, that allows the bacteria to germinate.
That makes so much sense.
A brand new, perfectly shiny nail pulled directly from a sterile box poses almost no tetanus risk. Conversely, a shallow scrape from a dirty, rust-free garden trowel can absolutely introduce the spores and cause tetanus.
And when you inevitably go to the urgent care clinic after stepping on that garden trowel, they give you a tetanus booster. A lot of people, myself included, assume that booster is a localized antibiotic, just cleaning out the bacteria from the wound.
It isn't fighting the bacteria at all. The tetanus booster is a toxoid vaccine. It contains an inactivated, harmless form of the tetanus toxin. It trains your immune system to produce antibodies that act like a security force. When the real spores germinate in a wound and start pumping out the neurotoxin, those antibodies intercept and neutralize the poison in your bloodstream before it can ever reach your spinal cord.
So the bacteria is still there.
Yes, you could have a wound that looks perfectly clean, with no redness or pus, but deep inside, a tiny colony of C. tetani is secretly manufacturing lethal neurotoxin. The vaccine ignores the bacteria entirely and just neutralizes their weapon.
So what does this all mean? When we zoom out and look at the toxin twins together, we see this incredible biological mirror. You have two indestructible spore formers. One gives you flaccid paralysis from food or drug wounds. The other gives you spastic paralysis from soil-contaminated cuts. But they are both driven by the exact same molecular mechanism, indiscriminately cutting those microscopic docking ropes in our nervous system.
And their relevance isn't relegated to history books, they are pressing modern concerns.
Exactly. With the massive surge in popularity of home food preservation and fermentation, the risk of improper low-acid canning is very real. Botulism is always just one bad batch of garlic-infused oil away.
It really is.
And tetanus remains a vital talking point in modern vaccine discussions because it operates under different rules. Tetanus lives permanently in the soil. It is not contagious from person to person. That means herd immunity does not apply.
Right.
If your entire neighborhood is vaccinated, it offers you absolutely zero protection if you step on a contaminated stick. It is a purely individual choice facing a purely individual risk.
This raises an important question, especially when we reflect on that taxonomy revelation regarding the mobile bacteriophages.
Right. The terrifying reality that the genetic blueprint for the most lethal poison on Earth is just a mobile piece of viral code floating around in the dirt, jumping between entirely different species of microbes.
It makes you wonder, what other perfectly calibrated neurotoxins are currently locked inside harmless, completely unclassified bacteria right under our feet? They are just sitting there in the soil, waiting for a viral phage to jump in and instantly transform them into the next toxin twin.
Now there's a thought to keep you awake the next time you're out working in the garden. Thank you for joining us on this exploration of the invisible, deadly, and miraculous world beneath our feet. Keep questioning, keep digging, and we will see you on the next deep dive.